Okay, everybody. I think it's 5:30, so I'm told we can start because the live stream starts at 5:30. We can't be early, but we can be slightly late. So, for those of you I haven't had the pleasure of meeting before, my name is Christina Victor. I have a number of roles at Brunel. Sadly, only one salary, but this evening I'm being Associate PVC for Research, and it's my absolute pleasure in that role to be able to introduce and chair some of our inaugural lecture series, which honours and celebrates colleagues who've been awarded promotion internally. So it's for those of you who are not in the academic world, it's much harder to get promoted in your institution because people know you if you apply from outside. So that makes Amanda a very special person, because her chair in biosciences is a reward for her contribution at Brunel over a period of years. I won't say how long, you can do that bit. And Amanda didn't start off at Brunel and did her degree in biosciences in Cardiff. She didn't stay in God's own country, but felt the need to move to Sheffield, where she did her PhD. Then did a number of postdoctoral posts at Sheffield and then at Royal Holloway, and arrived at Brunel as a lecturer. And during her time with us, I think Amanda's academic journey has been exemplary, and I think an inspiration to all of us. Obviously, she's an excellent researcher in her field of cell biology, particularly thinking about the link with cancer. So perhaps when we think about health services, we think about cancer, we think about the medics and the randomized controlled trials and therapeutics. But Amanda is one of that really important group of basic scientists who look at the mechanism and causes that enable us to develop treatment. So she has, of course, an impressive research career. She's also a very good teacher, so has a very strong educational profile, and is also perhaps one of the most collegial people on campus. So I've got on my notes here a list of the leadership roles that Amanda has executed with great skill and effectiveness during her time with us. But frankly, we'd be here all night if I listed them all. I think I will hand over. I just want to say one more thing. So academics are interesting people, and Amanda is an interesting academic, great researcher, good teacher, absolutely fantastic educational leader. But we all have a backstory, and I do hope Amanda will share some of it with us. I think she might have something to do with balancing. Are you going to mention your balancing skills?
Maybe at the end.
Yeah, maybe at the end. So I'll tantalise you with that. What she does when she's not spending her time doing research, inspiring her colleagues and teaching students, and generally leading us to the promised land. So it is my absolute pleasure to be able to welcome Amanda and invite her to present her inaugural lecture. Over to you. Oh, I was so enthused about Amanda because, as you can see, one of the pleasures of my job is to be able to see people progress. But I forgot to do the shameless promotion. So if you like tonight's lecture, come along. We have two more coming up that you can book for, and you can come along and then you can compare and contrast and rate this term's inaugural lecture. Okay, Amanda, the floor is yours.
Thank you very much. So just one before I start, a few more sort of housekeeping notices. We had Nana's as the closing inaugural from last term, and we heard from his band, we heard him sing. I was at my dear friend in collaboration, Alessandra's inaugural last year, and she sang at hers. For those of you that know me well, you will be relieved to hear there is no singing tonight. For those of you that don't know me well enough to sing, just rest assured you really don't need to. And Sergit's not here, unfortunately, but at the end of Nana's talk, she asked him what was, the most important part of any outfit, because clearly none of us can outdo Nana in the style stakes. And he said shoes.
Yeah.
So, my trainers have been replaced by my heels tonight. So housekeeping done. I'm going to talk a little bit about where I grew up, my life, a bit about life journey and career journey, and touch on a few highlights along the way. I am a breast cancer biologist. I do cell signaling research. I have tried not to put too much nerdy techie science in. I've got 1 or 2 slides just because I think it would be remiss not to. But for the non-scientists and for those who aren't familiar with when we say cell signaling, what do we mean by that? So we've got a picture of a cell here. Not actually not showing up. Oh, okay. Okay, cool. So we've got a picture of a cell here drawn in cartoon form. And around our bodies, we have loads of different growth factors, and they direct cell function. And to do that, they bind to receptors that are on the surface of the cell. And that then stimulates proteins inside the cell to initiate a chain of events that then direct cell behavior. So when I talk about cell signaling, that's what I'm talking about. It's not as simple as a single pathway that's up on the screen. It looks a little bit like a sort of spider's web network because there are a number of different signaling molecules, and there are hundreds, if not thousands, millions of receptors on cell membranes. So it's quite complex, but we'll keep it nice and simple. But before we get into that, we'll go back a bit. So I grew up in North Wales, in a small village, just not too far away from Wrexham. Neither of my parents has a university degree. I don't think we've got any students in the audience, but just in terms of what your background is doesn't necessarily limit what your potential can be. And when I was about 11 or 12, we swapped the mountains and the daffodils of North Wales for the windmills and the tulips of the Netherlands. So we went from living in a relatively small village, I think I looked the population up fairly recently, and it was around about 7,000, to living on the outskirts of The Hague. So quite a big culture shock in terms of just the sheer volume of people, but also the diversity that you encounter moving from somewhere so small to somewhere so big. We stayed in Holland for 3 years, and then we moved back to North Wales, which was where I finished my schooling. So I did my O&A levels back up on the North Wales coast. And I called this the unplanned career because my original plan was I was going to be a vet. That was the only bit of career planning that I've done, really. And as you can see from where I've ended up, that's not really how things have turned out. So my O-levels, I'm that old, I've got O-levels, not GCSEs. My strongest subjects were maths and French. Yeah, so things could have been very different. But I wanted to be a vet, so that meant I had to do science A-levels. So I did maths, biology, and chemistry. Of my A-level subjects, maths was by far and away the best. Biology was by far and away my weakest subject. So the irony of now teaching biology is not completely lost on me. But what I came to realise in the sort of second year of sixth form was I probably wasn't going to get the A-level grades I needed to get into veterinary school. I'd done loads of work experience. I'd got between 150 and 200 hours of work experience. I sat down and roughly did a mental tot of how much I thought I'd done in the run-up to applying for university. And I'm a little bit of a realist. And I sat there and I thought, well, if I'm not going to get in, because I'm not going to get either the predicted grades, so I won't get offered a place, or realistically, am I going to get 3 straight A's, to which the answer was no, I'm probably going to end up doing a science-based degree somewhere. And because I'm quite stubborn, I decided I wanted a choice in what that science-based degree was going to be and where I was going to do it, rather than being allocated somewhere through clearing. So fast forward, I ended up in Cardiff. I knew you'd approve.
Yeah.
So I did honours degree in biochemistry. I particularly chose a biochemistry syllabus that had not too much biology in it. And I think here starts a story of doing that extra bit and having that itchy right hand. I'm now getting to the stage in my career where I need to learn to sit on my hands in meetings, because if people are looking for volunteers to do something, I'm there putting my hand up. But it has served me well over the years. So I became a student rep and had my first encounter sitting in a student experience committee, trying to explain to the chair of the student experience committee that actually it was his lectures we didn't like. So, yeah, a skill that has served me well over the years. And at the end of the second year of my degree, we had a 2-week practical in the lab, which I absolutely loved. And what I should have said is my plan had always been that I was going to go and do biochemistry, I was going to get my 2-1, then I was going to go and do veterinary medicine. But I really, really enjoyed being in the lab. So I then decided that 2 weeks was great, but 3 years, I wanted to do science. So I went and did a summer studentship at the Heath Hospital, looking at how collagen is produced in wounds, depending on whether they're acute wounds, so things like surgery, so where you physically cut the skin, and chronic wounds. So at the time we were looking at piles. Thankfully, sample collection was not something I had to do, but the samples came to us, so that was good. And I had a number of other part-time jobs as well. And I said, well, in the brief, we were asked to talk about our challenges and we were asked to talk about the lessons learned along the way. If I'd given more thought to this talk, I probably would have subtitled it, "Because You Can't Trust It to a Girlie." And working at the Heath that summer was the first time I heard that. For the biologists, it was 1992, PCR was new. Now it is a bog standard technique, we don't think twice about it. But it was new, so you couldn't give it to a girl to do because you couldn't trust them with it. For the non-biologists, it's the end of a 3-stage process. And the bit that I always hold dear in my mind was I was working with a team of other students and we were allocated a stage of the process each. I got stage 2. But I'd messed stage 2 up. Stage 3 was never going to work, whether it was a girl doing it or whether it was a boy doing it. So that is something I always hold in the back of my mind. But the lessons that came from being in Cardiff in those years was to be open-minded, because I could have been very fixed and I was then going to go off and do my veterinary degree, but because I enjoyed something, I decided to explore it a bit more. So flexibility is quite important. Get involved in stuff. So that student rep, that extra bit. Be nice. So in that lab, there was a technician called Steve and 2 other students that I was working with. Darren, who was one of the other students, I met in a bar in London accidentally about 10 years later when somebody introduced me to their boyfriend. And he said, "I know Amanda." And I thought, I know that voice. So you never know when people are going to come back around into your lives. And despite the fact that we were looking at wound healing, Steve has now gone on to be a breast cancer biologist, and he heads up the Cancer Biology and Therapeutics program at Cardiff University, where I was their critical friend on program design, but I'm also now the external examiner. So, from being an undergraduate student, we now work quite closely together some 30 plus years on. And in terms of not being able to trust it to a girlie, yes, you have to take on board feedback throughout your career, you have to act on it, but people aren't always right. So I then went to Sheffield to do my PhD, and I chose Sheffield because, well, I applied to Sheffield because the project sounded interesting. I wanted a project that was going to give me an opportunity to learn skills. The reason I actually ended up going to Sheffield was, out of the places I applied to, they were the only one that offered me a place. So, a certain amount of realism there. I've put these slides up. These are slides of stained cells. It doesn't matter what we're staining them with. But now we have cameras attached to microscopes. We stain our slides. The camera takes the picture down the microscope. It automatically appears on the computer. It can be edited into the format that you need for a figure, and life is easy. Then we had a camera attached to a microscope that had 35mm film in it. And the fluorescence was really, really weak. So you couldn't spend time looking down the microscope to see if your cells were stained or not. You just had to take the pictures and hope, send them off to be developed. And then when they came back, you'd have a chance to look and see whether or not your experiment had actually worked. And then we had the joy of having to cut panels out and then I had to stick them into my thesis and then put them through the colour photocopier. And you think about image manipulation now, even on your phone, how simple it is. this was PhD standard image. Well, there was no option to do image manipulation. It's like, well, you've got the print or you haven't. What was really depressing was then if they didn't print, you know how you used to send your holiday photographs out and if there was a blank negative, they wouldn't print those. So I used to have to get them done on the 1 hour printing service to make sure that they printed all the blank ones because they were my negative controls and should have been blank, but I needed a picture of blank to put in my thesis. And then partway through my final year, my supervisor left. And this wasn't a period of time where you had second supervisors. We have research development advisors as well now. Students weren't particularly well supported in the lab. So I was pretty much left to fend for myself at the end of my PhD, which to a certain extent suited me fine, because it then meant I got to do all the experiments he didn't want me to do because he wasn't there to say no. I was basically able to crack on. But it did teach me a lesson in resilience and independence. My PhD years were really tough, and that's conversation for a glass of wine rather than for something that's going to be recorded, and I'm quite happy to chat about that afterwards. But I did, several years later, manage to get a paper out of my PhD, which was good. But then, obviously, finished my PhD. What next? Well, I needed a job. I also wanted to stay in Sheffield. Can't think why. Ice rink.
Nice one.
Yeah.
Nice.
Who knows? So I applied for a postdoc position and was fortunate in actually getting one. What I should also say, because there is a bit of a theme, and I didn't say it on the previous slide, is, note the 1970s and 80s concrete. Got a little bit more on the Hallamshire Hospital. So clearly I was enjoying my love affair with concrete, so I stayed and worked at the Northern General in Sheffield, which is also quite concrete heavy, and also at the Hallamshire for a couple of years. And I took a step sideways from doing cancer biology then. So my PhD had been very cancer biology focused. We were looking at colon cancer. I went to work for Holloway. He was a great person to work with for my first 3 years as a postdoc. And we were looking at the effect of growth factors on the enlargement of the prostate that happens with age. So for the biology bit, this is the bladder, and the prostate is a walnut-sized gland that sits underneath. And you can see you've got a nice open tube with the urethra so that the urine can flow. With aging, the prostate grows bigger and it puts pressure on that tube and it closes it slightly, which makes it harder to go for a pee. So we were looking at the role of growth factors in that and what they did, how they manipulated the tissue to be able to see what therapeutic interventions we could put in place. And the contrast I found working in that lab compared to my PhD was huge. I can still remember going in on my first day and sitting in the office and being handed a copy of the grant, an order pad, and it's like, right, plan your experiments, order what you need, crack on. And that was so totally opposite to what I'd experienced in my PhD. And it was like, oh, so there is another way to lead a lab. There is another way to look after people. But I still learned a lesson in resilience, because although I was in Sheffield for 3 years as a postdoc, I had 4 contracts in that time, the shortest of which, because of funding, was 5 months. And we had the conversation of, yay, you've got another 5 months' money. And I was like, yes, but I still need to look for another job because in 5 months' time, I don't have one. Probably should point out, I also got married while I was in Sheffield. We look young there. I did move resilience slightly further away from the photograph, just so it didn't look like the two were linked. I can put it back. Met Gordon, we got married. Gordon had been offered, if they could get the funding, a postdoc job in Australia. That, for varying reasons, didn't come off, and he ended up moving down to work in Surrey in marketing for the pharmaceutical industry. So I said, although we've got funding then in Sheffield for me to stay for another 3 years, I said, well, okay, if you're not going to move to Australia, I guess the least I could probably do is manage to move as far as Surrey. It's not quite as far as Australia. I think I can probably move that far. So I started looking for another postdoc job. And that's what ended up bringing me to Royal Holloway. And I applied for the job at Royal Holloway based on the skills that I had. The other bit, yes, the right group head makes a difference. So, whereas my PhD had been a very different experience, working with Colby He is such a lovely person. Really good liking of fine wine and good cheese as well is one of my take-home memories from those 3 years. But, he really gave me a little bit of confidence at a time when actually I probably didn't have very much. And that was really, really valuable. So I then moved to Royal Holloway, which is actually the start of the breast cancer story. So Christina said last week, you said, oh, I'm really interested to know what made you get into breast cancer.
Yeah.
I was looking for a job down south, as you do. It was an interesting project. Mark, who's a lab head, was, again, a really, really good boss to work for. But he was very, very heavily molecular biology focused with his experience and was looking for somebody who could bring a lot of cell biology skills. So the fact that I'd previously been working in prostate and in a non-cancerous condition didn't actually matter. What he was interested in was my cell biology skills. He didn't like doing cell culture. So the fact that I said, yeah, well, I've spent 7 hours a day in cell culture for the last project for what feels like a lifetime, was great because that meant I was in a good position to get the job. And we started to look at this enzyme that's called BRK. So BRK, it's breast tumor kinase. We refer to it as BRK because why wouldn't you? And we were interested in knowing what it did in terms of breast cancer development. So we were looking to see if you switched it on or switched it off, whether the cells divided more quickly, whether the cells divided more slowly, whether they survived in places where they shouldn't necessarily. But again, this sort of itchy right-hand syndrome crept in. And I sort of volunteered myself to be the research staff rep on the management board in school. There was an opportunity to get involved in doing some teaching, so I did that. And Mark and I also wrote a number of grants together. We had a lot of opportunity to write grants, so it's one of the reasons why I ended up being there for 6 years. I was there originally on a 3-year project, and then I wrote a grant for the second 3 years that I was there. We also, between us, wrote grants for another postdoc to come in the lab and work on the same enzyme, but she was looking at it in skin. I'll come back to that in a bit. And we also did some work on leukemia as well, which we've put grants in. While I was at Royal Holloway, the Institute of Learning and Teaching was formed, so I became an associate member of that. And there's probably only a few of us that are old enough to know that that was what Advanced HE were called before they were called the Higher Education Academy. They used to be called the Institute of Learning and Teaching. So I had associate fellowship then. And just in case you think, the fascination with concrete has gone, this beautiful chateau building. Nah, that's the biosciences building at Royal Holloway. So, I couldn't really escape the sort of the concrete jungle. And in 2002, my daughter was born. I think she knows I've used this picture. I did ask her for some pictures, but it wasn't on the list, but it's one of my favorites. So I pinched it. It's one of the few times she's looked cute. So, you have to use them where you can. And I had a great 6 years at Royal Holloway. It was probably one of the sort of the best 6 years of my career, actually, because I got to got to be involved in management board meetings. I got a bit involved in teaching. I got to write grants, I wrote papers, I did loads of lab work. I had no accountability for any of it. So it was kind of doing everything, but without necessarily having any of the responsibility, because that all sat with somebody else. It was great. Really enjoyed it. Working for Mark was great. He became known in our lab by one of the guys in the lab. By her boyfriend used to refer to him, your boss, the man with the brain the size of a planet, because he had so many ideas. It was difficult to keep up with them. So, Ali, I don't know if you know this, we used to triage his ideas. I told him this fairly recently. He hadn't realised we did it. So he'd say, oh, I've been reading. I think we should do this. We're like, okay. Yeah, right, okay, I'll bear that in mind. When it got to the third time of him saying, have you looked at this? I can remember Jo coming into the lab one day going, oh, Mark's mentioned, I can't remember what it was, but she said, for the third time, that means I'm going to have to do it now, isn't it? And that was, in order to be able to filter some of the stuff that he wanted us to focus on, we triaged. But he really pushed us. And that for me was really, really good. And you look at the stuff that I was able to do while I was working in there for 6 years. That was everything I needed to get experience to be an academic. So the extra stuff helps, as long as it's not too much. Sit on that hand. But also, so does the right support. back towards the end of 2005, 2006, I wrote a personal fellowship for what would have been a 5-year project for me and some technical support to what was Breast Cancer Campaign. They'd launched a new fellowship scheme. And although mine scored well, it wasn't funded. But what they did do was give me an opportunity to resubmit it as a 3-year project grant, which was great because it meant I had to cut 5 years down to 3. So I just cut out all the bits I didn't like and then resubmitted the 3 years that they did like. And in that gap, the job at Brunel for an academic was advertised. So I applied because I'd got the experience, got the role, but it then meant I had the grant was awarded. So I then had a grant to employ a postdoc on when I arrived, which was great. We published quite a lot of papers at Royal Holloway, a number on this molecule, Burke, and then a couple on leukemia as well. And then I've highlighted this one in green because this is work that Joe started, but looking at the role of Burke in the skin. And in normal biology, Burke's not really found that much in the breast, but it is found in normal skin at quite high levels. So one of the things that we were interested in knowing about was what is it doing in the skin in normal conditions that we could then manipulate when it's switched on in a tumour to be able to control tumour growth. But also, why is it in the skin in the first place? And it's one of these more difficult molecules to work with. And Annie, I know you're online. You will hate me for introducing you to this molecule because in normal cells, it stops them from dividing. In tumour cells, it makes them divide really, really quickly. So it has completely different functions. So we started to think, well, what is it about skin and breast tissue that are potentially similar that means this molecule gets switched on? So this is the geeky science slide. Said there'd be one. So there's a lot of literature that suggests if you take vitamin D, it can reduce your chances of getting particularly breast cancer, but also prostate cancer. And there's lots of studies showing that the nearer you live to the equator, the lower your risk. And it's, there's obviously diet and lifestyle factors, but also, exposure to vitamin D is believed to be something that plays a part. And clearly, we're not going to take cells from the lab and shove them out in the sunshine. But what we can do is we can treat them with vitamin D, because you can buy synthetic vitamin D. So here's the biology bit. So when breast tissue develops, you end up with these sort of duct-like structures, and then you have a lobe at the end. And these purple cells eventually die, so you have a hollow lobule at the end. But these grow in from the invagination of the cells that are on the outside of the body during embryonic development. Cells that are on the outside of the body during embryonic development are also going to be the same cells that are going to sort of form your skin. So we were hypothesizing that perhaps there are subtypes of breast cancer that actually, remember is too strong a word, but share evolutionary features of the cells that they once came from, which is why they resemble skin to a certain extent. And when we have Burke at high levels in breast cancer cells, they also express, so they display markers and features that we would associate with skin cells. So there's definite similarities there. And the reason I've got this sort of purple sprouting broccoli, and apologies now because you're never going to look at broccoli in the same light, is that in the breast you have loads of these sort of ducts with the lobular structures at the end that all link together, that form, and the only way to really describe them if you look at them down the microscope is like sprouting broccoli. So you have the stems, and then you have the little fluffy bits on the end. So these are the fluffy bits, and these are the stem bits. Next time you're having a healthy tea and you're having broccoli, you can say, I now know what breast tissue looks like on the inside. And what we showed was that where you've got high levels of this protein Burke and our skin markers, it predicts whether or not cells will respond to vitamin D. And that was quite interesting because the literature is quite confused on whether vitamin D is chemopreventative, but also whether vitamin D has any therapeutic benefits, or can you use it as a treatment? But it depends what cells you're treating. So we think that if the cells have Burke, and if they have the keratin, the skin markers, then they will respond. If they don't, then they won't. And that's why the literature is quite vague. So this work I started in Mark's lab prior to me leaving in 2006. We eventually got it published last year. So if you guys can buy that, the development of the breast is a bit like the skin, I'm going to send you lot my paper to peer review next time, because trying to get that concept across to reviewers was incredibly difficult, and it's taken us a huge length of time and a lot of tenacity. So a lesson learned is to hang on in there, because if you've got the courage of conviction, stuff will eventually come right. And then just before I left Royal Holloway, Alessandra, who's in the audience, joined Royal Holloway. She's a plant biologist. And she had, and I'm going to use the word crazy. Yeah, you don't dispute that, do you? She had this crazy idea. If you take bits of plant, and depending on what you've treated the plant with, and you lob them into your cell culture, so your breast cancer cells, cell behavior will be changed. And you might be able to stop the cells from proliferating. Now, all the biologists in the audience are going, because we're chucking plant cells in. For those that aren't scientists, as biologists, we spend most of our lives trying to avoid contaminating our cells with anything. So the idea that you would then take a bit of leaf and chuck it into a flask of cells was completely alien. So I think I was interested but resistant for a while. And then she eventually wore me down. And I was like, do you know what, I'm just going to have to do this, if only to say it's not going to work. She sent me various plants, along with your researcher, who spent ages cutting out 3mm leaf discs in the hoods out of plants, putting them into culture. And would you believe it? We actually found there was a difference. So plants produce this hormone belonging to the jasmonate family. And we showed that actually jasmonates have the potential to selectively inhibit breast cancer cell proliferation. They didn't have the same effect in normal cells. We've got a bit of a differentiation effect there. So we started this work probably about 2008, maybe, probably. We managed to get it published in 2021. So again, another of these slightly wacky things that we had to go from journal to journal, because if we went to the biology journals, there weren't enough replicates, there weren't enough cell lines, but actually completing a 96 well plate with 3 leaf discs per well takes a huge amount of time. We went to the plant journals, they weren't interested because there was too much human biology in there. So eventually we found somewhere, the New Phytologist, which is a relatively high-ranking plant journal, did take the work. Alessandra ended up in the Times, so this is the image that I've taken from that. We also ended up in the Daily Mail. So it caused a lot of interest. But it's worth it. Sometimes you may as well just say yes, because in the long run, you're going to anyway, so you may as well get it over and done with and do it quicker. Mad ideas do pay off. But again, you need to have that self-determination and belief that actually what you're doing is going to produce the results, and you are eventually going to get that paper out, because it is a long, hard slog. So then we move on to the Brunel years, because clearly, I've had enough of being in a concrete building, but too much prettiness. So I need to reaffirm my love of reinforced concrete. So back at Brunel. So I joined in 2006. It's my first academic post as a lecturer. We had 3 years probation back then. So I hadn't finished my probation, but I was asked to be the learning and teaching coordinator for the division, which is effectively the equivalent of what is the program lead role now. I volunteered myself to be Associate Deputy Head of School in 2012. And then when we restructured in 2014, there were a whole load of new roles produced and advertised, and somebody came and said, "Oh, we've got an Associate Dean Quality Assurance role. You'd be great for this." And, having done Chair of Board of Studies as part of the Programme Lead role and various other things, that itchy right hand, it's like, yeah, okay, I'll apply for that. Not expecting to get it. And I did. But it took from joining in 2006 to actually being promoted to senior lecturer in 2015. That was a long, hard slog. A lot of us applied in 2014 and was rejected. 2015, I was successful. Part of that was due to the promotion criteria changing. Part of it was due to my daughter getting a little bit older. So you're able to do that little bit more and get involved in that little bit more in things. So did the Associate Dean role for 3 years. And at the end of the 3 years, they were then making all the roles 4 years. So I was asked if I wanted to stay on and do a 4th year. And I said, no, I'm going to apply for research leave because my research had taken a bit of a battering during those 3 years. And I thought I actually need to focus on, if I want to get promoted to reader, I'm going to need to do some more research. So that's probably one of the few bits of planning I've ever done, because I thought ahead. However, unbeknown to me, head of biosciences left. So I was appointed head of biosciences on an interim basis in the middle of 2017 and decided wisely, madly, delete as appropriate, that I was going to apply for the role. So I applied for the role of Division Lead for Biosciences and was successful. 2017 was the academic year where we grew our student numbers by 50%. We were told we could have some more staff, which was great, but obviously we had to advertise, we had to recruit. It was going to be time before those staff arrived. What I hadn't realised when I applied for research leave was 3 of my colleagues had also applied for research leave. So potentially we were 4 members of staff down because of the increased teaching and 4 members of staff down due to research leave. The other 3 were really accommodating and moved when they were doing their research leave around teaching. I chose not to take mine because I felt I couldn't 2 weeks into the role then say, bye, see you next year. Here's all the problems. It's just not how I work. So I actually didn't take my research leave. And I think at that point, I probably committed myself more to an academic role that involves collegiality management and leadership more than research. I still do research. I still bizarrely like getting into the lab to split cells. I feel like I've been let out to play. I think my group, Mal certainly would probably prefer that I didn't because I just get in the way and I don't know where anything is anymore. It reminds me when I was at Royal Holloway and Mark used to come into the lab after a while and it's like, oh, he's got his lab coat on. And we'd arrange things so they were easy for him to find so then he didn't start messing things up. And then between 2015 and 2022, I had relatively quick promotions to reader and then to chair in 2022. So it took me 9 years to do the first step and then took me 6, 7 years to do the next couple. In 2020, I'm not going to go into the reasons why, we all know why, there was an interim deputy dean post available, so I took that on, thinking that would be good for my career development. What I didn't know in January 2020 was what was going to happen in March 2020. So there's nothing like stepping into a new role and making it your own when you've got an international pandemic to deal with and thinking about how you're going to lead the college through teaching. And thankfully I was really well supported by Claire in her Vice Dean Education role at the time. And I did that role for 3 years, and then with the restructure that we've had, the Associate PVC Quality Assurance role came up in 2023, which I applied for. And although it's got quality assurance in the title, the bit that I really enjoy is the access and participation work and the planning and thinking about what we're going to do for underprivileged students and students who haven't had the same privileges as we have through our careers. And that's probably one of the things that drew me to that role. One of the things Gordon and I have, we have quite heated conversations and evenings, don't we, about various aspects of it. And it's a bit that I really, really do love. Still publishing, so still working on Burke. It's starting to become a molecule that is blighting my life. But I still have faith that at some point there might be something in the lab as a co-therapy that might be based on it. But I've also developed an interest in other molecules that are involved in breast cancer, and so ACBD3 was one. I'm not going to talk about that particularly, but one of my PhD students was working on. So while I've been at Brunel, I've had quite a few external roles, particularly relating to teaching, but also relating to research. So got professional memberships of the relevant associations, as you might expect. So the British Association of Cancer Research, and substitute European and American for the E and the A and the other acronyms. And I'm a fellow of the Royal Society of Biology. Because of the fellowship application I put into Breast Cancer Campaign and then the successful grant application, I was asked to join their advisory board as a grant reviewer, which I did from 2008 through to 2012. And then I have passed on the baton very ably to both Paola and to Mike, as they have succeeded me in that role. It's a good experience. It's good for the soul, isn't it? Possibly. But while I was on the advisory board, we had a gap analysis, which was effectively a massive paper looking at all the gaps in breast cancer research. And I was a deputy group lead for one of the groups that was the work was divvied between. I then joined the BACR Executive Committee in 2016, and I was nominated for that by Steve, who I met in the lab when I was an undergraduate student back in 1992. So that, be nice to people, treat them with respect, be professional is really important. So I joined the BACR Executive Committee. I served a year then on the committee and then became Treasurer-elect. I then managed to do 6 of a 5-year term. Don't quite know how that worked out as Treasurer, partly because of pandemic reasons, and we weren't able to have a change, so I wasn't able to step down. And then I am just coming to the end of editing a special edition on cancer cell signalling for the journal Genes. Sort of my external teaching activities are focused on a collaborative award for the teaching excellence. Biosciences, there were a lot of us involved on that. I'm nominated lead on paper, but it's not a sole venture, it's very much a team effort. David and Sabrina will attest to the number of talks they've done around the country, spreading the word. And between us, we've spoken at a number of meetings led by Advance HE, National Teaching Fellow Conferences, Royal Society of Biology meetings. We edited and published a book, and that stemmed from a conversation with David when he was block lead for a cancer cell signalling module at master's level. And it's like, we haven't actually got anything where all the resources are together that the students need. We direct them off to papers, but it would be great if we could have a book. So, yeah, okay, well, we'll do something about that. So David wrote a chapter, Manos has done a chapter, Gudrun's contributed a chapter to the book. So managed to haul a few friends together, both local and international, to write in that. Done my fair share of external examining, been a critical friend for programme development at other institutions, and I am now an external reviewer for the Advance HE Collaborative Awards for Teaching Excellence. So we've just done our first round of reviews. So it's probably about time for another lesson learned. It doesn't exist. To quote Jean-Luc Picard, make it so. Yeah, we didn't have a resource. We went out and we produced one. And it was doing stuff like that enabled me to be able to make successful promotion applications. I think my sort of 40-ish minutes are kind of coming to an end. We've got a few slides left. I think I need to start with some professional acknowledgements. I've been extremely fortunate to work with a wide range of people and some really talented scientists during my career. It's been really, really, really inspirational. Said Alessandra from Royal Holloway is here. Mark wasn't able to make it. Colby has now retired, as is Dylan from UEA. Oxford, worked with Esther Hammond and Isabel Perez. Isabel moved to Hull. She's now at Manchester. Particularly Annie from Brunel. I think I need to owe you an apology rather than an acknowledgement, because I've introduced them all to the wonder of the molecule that is Burke and the difficulty that is in working with it. So you're very welcome, guys. And I've had the opportunity to collaborate internally with a number of labs, so we've had quite a few collected papers together, and we've got a few more still to come. I do, however, have the support team at home. And you can interpret support how you like. So I asked my daughter, could I have some pictures? So she sent me 14. So I chose a few, from the early years, She's a musician. She's not here today. She had an assessment in Southampton. Apparently it's gone well. She messaged me, obviously. So that's all good. My brother's not here today. He's on call. Apparently patients like anaesthetic when they have operations. So Michael, if you're listening online, I promised I'd give you a shout out. So hopefully you haven't been called into theatre. I asked my husband for some photographs that I could use. And he sent me that one. He then said, oh no, it's supposed to be professional and serious, isn't it? I followed it up with that one. I did find some nice ones though. You haven't seen one of the ones I've shown, so I decided I wasn't going to ask. So one slightly more recently, one a while ago, yeah, but I like it. But one quite recently, we're not quite so young, but we still do scrub up fairly well on occasion. Do have a few concluding slides, final lessons, so to speak. You have to accept the inevitable. I'm getting to a certain age where I use phrases, I find myself doing things. Sorry, Mum. I am turning into my mother. What I'm not certain about is who that's more terrifying for, me or for Gordon. But at the end of the day, I think the bit that it's very easy to forget when we're in academia, there will always be something else that needs to be done. There is always the next thing, the next grant, the next thing, the next paper, the next committee meeting to go to, the next thing that you need to read. Is that just occasionally we need to take a step back and remember that we don't live to work. We do work to live. So it is important to take a little bit of time and have fun. And to spend time with friends and family, the people that are important and the people that matter to us. To go and see the places that we really like, and you'll notice both from the people slides and from the places slides that water features quite heavily. Well, specifically for you, Christina, I thought you'd appreciate that. No, yeah, you can't. You have to be able to do your own straight line. So this is in North Wales on Colwyn Bay Beach. This is looking out over St Andrews, and then we're fortunate to have family in far-flung places, so occasionally we get to go and swim in tropical waters. But despite all that, I still prefer my water slightly frozen. John, if you're online, I don't know if you are, but he said, will there be an ice skating photograph? And that's probably the only one I like enough to include. There are no decent photographs of me in action, so you're not going to see any. And what's life without the odd cocktail or two along the way? And then finally, this is my final slide, I promise. I'm of an age. There is no proof of my stupid stuff when I was younger. It's not recorded on the internet. But, on occasion, you still need to get out and let your hair down. The lesson is you need to be careful who you do that with and where it's recorded. Because occasionally, photos come back round. So, out drinking cocktails with Sandra and Elizabeth, one of my cherished memories. And on that note, I am going to end. Thank you very much. It's lovely, isn't it?
Thank you, Amanda. Nice pictures of water. I'm a very keen swimmer, so everybody has to put a slide in with water
and our paths occasionally cross at Spectrum on Monday mornings, don't they, for different reasons.
What time is that? 5:30?
Yeah, usually.
Yeah. That's why we're usually asleep at this time.
Yeah. Why go in through the front door when you can go in through the fire escape because nothing else is open?
The swimmers go in through the front door, so we're obviously privileged. They don't send us in the fire escape. Very thought-provoking and interesting lecture, Amanda. Anybody like to ask? Straight out the blocks. I can finish my sentence. Do you need a?
very dynamic tissue, breast tissue. And obviously, as women it changes in the cycle. So as I say, it's dynamic tissue. It changes in the menstrual cycle, when we're pregnant, when we're lactating, the menopause, and then we can no longer defy gravity. So have you looked at all the aspects of that? And then the second question is, why is there an affinity for bone metastases from breast cancer?
So for the first part, we haven't specifically looked at every single stage during development, but others have. And in relation to the bit that I work on, there doesn't seem to be any correlation with switching on of the proteins we're interested in at particular times during development, which is why we have just focused on the cancer bit. In terms of why particularly bone, there's a lot of literature supporting sort of seed and soil hypothesis that there are certain things about the bone that makes it a very fertile ground for particular types of cancer, so breast cancer particularly, prostate as well. Others less so. And exactly what that is, there's literature suggesting certain aspects, but we don't have a definitive answer for all of it yet. But there's certainly a lot of drugs that are coming through from particularly osteoporosis treatment that are used to prevent tumours from spreading to bone.
Cherry at the back.
I have to apologise, I think so late. I was programmed at 6 o'clock. So, sorry everybody. But
Did you not get the memo?
Really not. No, missed out on that one. So, but anyway, delighted to have got it. I just really want to talk about a serious question.
Yeah.
Amanda, you and I very
We have. Do you know, I haven't looked at them in a little while. So this stems. So Cherry and I both started at Brunel around the same time, and we were what was the PG cert for academic development back then, and we were having a conversation over coffee. And your son was doing either his GCSEs or his A levels, and he was struggling with quadratic. And I went, "Oh, I loved them when I was at school." So yes, it's a standing joke every time we meet.
Excellent. I think there it is. You need the microphone?
Thank you so much, Amanda. That was absolutely fascinating. I'm speaking as a breast cancer survivor and also as an arts and humanities scholar. So as a breast cancer survivor, I just want to say thank you for whatever you did, in whatever way. Maybe I owe you my life.
Absolutely.
And as an arts
unlikely.
and humanities scholar, I just want to say I was so touched by when you made that connection, and I obviously have no idea what I'm talking about, right?
Yeah.
But the little plants, the 3 millimetre little bits of plant in the cell thing, and then it taking so many years to get that published, it was so poetic. Like, it was just beautiful to hear that. So thank you so much. But my question is, should we all be taking vitamin D, and if so, how much?
It's a very good question. And I'm going to have to go with the stock answer on that is it never hurts to take additional vitamins, but not to exceed recommended allowances, because too much vitamin is bad for you. And actually, if you have too much vitamin D, it can really then mess with your calcium metabolism, because vitamin D is required for stabilizing calcium in the body. So if you overdo it, then you need to make sure that you don't. So I wouldn't necessarily suggest that everybody goes out and has double doses of vitamin D by any stretch of the imagination, but a little would be good.
Proper academic answer.
Yeah, it's the same with eating your greens in moderation with your chocolate, isn't it? That balanced diet. If I've got an apple in one hand and a bar of chocolate in the other, does that make it balanced? Yes.
Joe. I guess I'd just like to say something about you as head of the business. You always knew the ins and outs of all the rules.
if there were new ones, you always were updated, and you had it right there in your mind at all the meetings, and you were always completely and utterly fair to every student, and you always looked where you could possibly help them and the staff. So thank you very much.
Well, thank you. That's really kind.
You don't need to do a Google search. We just ask Amanda. Yeah.
Yes.
I suppose, what's next? Any predictions what the future years of your career may entail?
Well, that would require planning, wouldn't it?
Yeah.
And as we've discussed, I don't do much of that. I see what comes along, and my itchy right hand has served me well so far that when opportunities come up, I quite often find I've got the skills for them. So we'll see what happens.
Thanks, Amanda. I was just wanted to talk a little bit about, or ask you about the starting point of your career. And you said you were the first in your family to go to university.
Yeah.
And there may have been periods of self-doubt and so on. Were there particular role models or things that happened that gave you that self-belief and really sort of started you on your academic trajectory?
I think, our parents were very keen that both Michael and I went to university. I can remember the conversations when, Michael, if you're listening, I'm sure you can remember them too, when you were having second thoughts about whether or not you were going to go. And it was, we were very fortunate in as much as, the conversation wasn't really about whether we would, but what were we going to do. And that was very much, I think, an expectation that was set for us very early on, because my mum left school at 16 because her father was ill and she needed to work. My dad grew up in Ireland, started a university degree, had to leave again for work reasons and came over to the UK. And I think always struggled throughout his career because he didn't have a degree, whereas, a lot of his colleagues did, and it made getting promoted very difficult. So I think that was always a driving force. It astonished me when I found Mum's physics A-level certificate, because I was like, you did physics? I thought that was too hard. I wasn't going to do physics. But in terms of people who inspire me, I think certainly now, I think my daughter is somebody who really does inspire me. She's had a lot of challenges in her life. And for somebody who can't hear, music would not necessarily be a first choice career. And she has never let that get in her way. And she's always been very focused and very driven on what she wants to achieve. And I think, that is certainly something to draw inspiration from.
Lovely, thank you.
More questions? In which case I'll ask mine. Would you rather have been a vet?
Well, I don't know, because I don't know. But, there are times when it is peeing down with rain and you think, do I really want my hand shoved up a cow's backside?
You don't do large animals.
No. Well, the practice I did my work experience in was the practice for the zoo. So we had all sorts of things in. So we had a penguin that couldn't swallow in the vet surgery one. The sort of stuff that you normally wouldn't get to see, perhaps as a vet, let alone as somebody just doing shadowing experience. So that was really fun.
Oh, sorry. I was looking at the wrong side of the room.
I just wondered if you wanted to say a bit more about your current role. So you said you're supporting students to access different things, but I don't know exactly what that means.
So I'm working with Claire and with Emily, who's up at the back. We're putting together the university's access and participation plan. So one of the requirements for charging the fees that we do is that all universities need to have a plan that's approved that says what we are going to do to help students from underprivileged backgrounds succeed while they're at university. And it covers a wide range of things, from financial support to mentoring to career progression discussions. I've gone blank now, Emily. I'm going to put you on the spot. We have about 13 or 14 project leads working on projects that are targeted at specific groups, and we spend a reasonable amount of money, sort of in the order of millions each year on that sort of work. Yeah. Yeah. So hopefully, the idea being that we will get to a point where over a period of time where no matter what somebody's background is, they have every opportunity to be successful and to come out with the best degree that they possibly can, no matter what disadvantages they may have in their life. That's the overall aim.
We all done for questions? In which case, I think we can thank Amanda again for a very informative and entertaining lecture, and I'm sure she'll be happy to talk to you outside where we have refreshments. So, thank you, Still missed. Yeah, in your